And like it's really really wrong because Calvin cycle if you remember the coupled reaction, so Calvin cycle is coupled to the light reaction. So if there are no, and I quote, light reactions, or when there is no electron transport chain happening on the granium, and then if there is no input from the light reactions, then the Calvin cycle cannot happen. Okay, so both of electron transport chain and Calvin cycle are happening during the day or when plants have light. Now, I really enjoyed this part of presentation because this is where I got to ask you a lot of logical questions. And this is where, you know, it's where biology is asking chemistry, why is this happening? So you will see a lot of chemistry, and then, you know, in biology there is always a very good reason why. There are three main stages in the Calvin cycle, and these are carboxylation, reduction, and regeneration. Okay, so in the process of the carboxylation, this is where the carbon dioxide is being fixed or absorbed. And the process of the fixation of carboxylation is done by an enzyme called Rubisco. CO2 has to be attached via this enzyme who is fixing the carbon. So here it is, Rubisco fixing the carbon dioxide onto this molecule, which is called ribulose 1,5 biphosphate. And it's called ribulose 1,5 biphosphate because it's a ribulose sugar that has a phosphate group on the first and the fifth carbon. So it has two phosphate groups on the first and the fifth carbon atom. Then next step is the reduction. So this is where ATP and NADPH are being reduced. So this is where we are going to use that energy which we created in the light reaction to create those sugars. And then in the third step is the process of regeneration, where we need to have, when I say we, I mean me and plants, and I don't know why I always keep saying that, but when I say we, you know, I mean me and plants. But in the regeneration part is plants need to have enough of this ribulose 1,5 biphosphate so they can maintain in fixing the carbon. So you will see that this process of regeneration is extremely important to plants. This is the image that I found on Google. So when you Google Calvin cycle, this is what's going to come out. Here is the ribulose 1,5 biphosphate. So you see this is the phosphate, and then there is another one. Ribulose has how many carbons? We can count 1, 2, 3, 4, 5 carbons. So once Rubisco fixed the carbon onto RUBP, you're going to have an intermediate molecule which immediately breaks onto two molecules which have 3 carbons, and they are called 3PGs or 3-phosphoglycerates. And now one of those 3PGs will be converted into 1,3-biphosphoglycerate and then converted into G3P. So glyceraldehyde triphosphate will then go into regenerate ribulose 5 phosphate, and then that is going to be converted into ribulose 1,5 biphosphate. So what is nice about this image is that it's a very nice representation. The structure of the molecules is correct. However, you know, if you're a chemistry buff, I mean like a really crazy chemistry buff, you will see that there is something seriously wrong with this picture, okay? Because you will be like, okay, here I have 5, and now I have 6. Okay, so 1, let's say it's going to be transported out, and then I have 6, and then how do I come up with the 5 from here? Okay, something ain't right here, and that is correct. So if you were to count, the carbons, you will see that this image makes completely no sense. And that is because actually what is happening is that 3 molecules of carbon dioxide are being fixated, not 1 like in previous cycle, but 3. So even though this image is from an old book that I pulled out, this is the actual representation of the Calvin cycle. So when we have 3 carbons, we need 3 molecules of ribulose 1,5 biphosphate, or RUBP. Okay, so here you can see we have 5. So then here there are 15, so 5 times 3, that's 15. And when we add 3 more carbons, that's going to be 18, which are then broken, so see, into 3 molecules of 3PG, so 9 here and 9 here, so that's 18. And out of those, only 1, okay, out of those 9 which are being created, only 1 will be used as an output, meaning that only 1 will be used to create later glucose. Okay, so this will actually be exported out of the chloroplast and then assembled into glucose. And all the rest of the 5, so now you see you have here 15, so 18 minus 3, that's 15, so that's the regeneration right here. So only a very small amount of these trio sugars will be exported out for producing sugars. The majority of the 3PGs will go back. So what we can see here from this picture is first important thing is that 3 molecules of carbon dioxide are being fixed. The number of carbons in this cycle, everything is right. Okay, there is nothing missing. And we can see here that only a small amount of sugar will be exported out. The majority will be inputted into the process of regeneration. Now, this is kind of telling us something, and it's telling us that plants really, really pay attention in having a pool of RUBP. Because if they don't have enough of RUBP, they can have light, they can have water, they can have carbon dioxide, they can have all these conditions, but if they don't have enough of RUBP, then there will be nothing to fix the carbon dioxide too. So you see also in another cycle, sorry about me skipping this. So anyway, I will come back to that because sorry, my slides are not in great order, which you already know. So you will see later how important to plants is to have this pool of RUBP. You will see what they're trying to do. So let me just mention one more time what I mentioned at the beginning. Very important thing to remember is that glucose is actually not the first product of photosynthesis. It is this 3-carbon molecule, 3PG. Okay, so that's the first stable sugar that is being produced. Okay, so just a short reminder, please don't forget that because this is where you have to draw a line between high school and college level biology. Now let's talk about Rubisco shortly. I will talk more later about Rubisco, but Rubisco stands for ribulose 1,5-biphosphate carboxylase oxygenase. It is one of the most abundant proteins on Earth. It has a copper core, and it's called carboxylase oxygenase because it can fix both carbon dioxide and oxygen. So put a sticky note in your brain onto that because you will see what is going to happen with that later. Now, this complicated graph or structure, it's a series of biochemical cycles which we will come across very often. And this is where I was supposed to ask you, what is going on with this complex biochemical cycle. So right here you see there is RUBP, there is Rubisco fixing 3 carbons, there are 3 PGs, and then some of them are being exported, and then some of them are converted into some other chemicals, and then you hear there are like different types of sugars with different numbers of carbons. But one thing that you should be noticed, right, and I'm asking you to kind of think about it, is that all of them will, you see, like they kind of end up, if you were younger and then you have to play the game follow the lines, when you kind of follow all the lines with all these different sugars, they all kind of end up right here. So this graph is kind of showing you to which length plants are going to make sure that they have enough of RUBP. So they will convert all these different types of sugars with different numbers of carbons, with 5, 4, 7. So these are different erythroses, xyloses, so different types of sugars which they will convert just to have enough of RUBP. Another important molecule is dihydroxyacetone phosphate, or DHAP, which is kind of an intermediate molecule, either when you want to export sugar or if you want to convert that into something else. Like in this case scenario, what happens is that there is a pool of G3P, and if light is becoming dimmer and then night is approaching, then some of that will be converted into starch. On the second lecture, I show you chloroplasts with dark granules inside. So that's the starch from plants just doing their job so efficiently. So there is a lot of starch that is being accumulated and didn't kind of have time to be, exported out. The rest of those, you see, will be converted into different types of sugars, but eventually we really need to have those RUBP. This is just some math. So if you want to make sucrose, which has 6 carbons, you need to fix 12 CO2 molecules. Now, in order to regenerate RUBP, it's not just going to happen. There is a lot of biochemistry that's being involved in that. So there is some time that you need to kind of replenish the pool of RUBP, and that is called photosynthetic induction time. This very simple graph can show you something really amazing, and that is and that is the plant will only put a small amounts of sugars into converting them into complex sugars and reserve material, but the majority of those will be used for RUBP to have enough of RUBP, which I already mentioned. Now in the next lecture, I will be talking about Rubisco, and